F5 प्रेस कीजिए वहां पर।
जी सर, फुल है सर, इधर से फुल की है मैंने सर।
F5 प्रेस करो तो।
हाँ सर, F5 प्रेस किया सर। ठीक है सर। हेलो,
अभी भी वैसे ही है नंबर जी।
ये पीपीटी में आप कर सकते हैं क्या इसको? पुलिस पीपीटी कर दीजिए।
पीपीटी को अपने फुल स्क्रीन करना है।
अब वही सर।
नहीं, अभी भी कुछ नहीं आया नंबर जी, एक बार दोबारा करो।
ऑफलाइन वाले तो
हाँ, अब हो गया पितंबर जी।
थैंक यू सर।
हाँ, अब करिए।
The cross transferability of the wheat SSR markers to the polygonal verticillium accessions. And this is the polygonal verticillium plant in its natural habitat. You can see the plant in natural habitat along with its various isolated part, just like flower. And Polygonum verticillum is locally known as a Salam Mishri, and this plant is a native of the Himalayan region and found distributed from the height 1800 to 4000 meter above sea level. And you can see in the diagram the distribution of this plant in the in natural habitat of the northern and northwestern Himalayan region. And this plant prefers to grow under shady and moist places, and basically under the forest understory. And this species basically understory species, and the plant grows up to the height of the 1 meter, and generally consists of the unbranched stem, and it bears the three leaves which originate from a single central point of main stem. The plant permeates to the underground part, which is rhizome, and this plant is has many therapeutic uses. And the rhizome is used to cure the stomach diseases by the folks, and used as a tonic. The different population of polygonal verticillum are under the high anthropogenic pressures, which is causing their diminutions. And the plant species of immense importance, its germplasm remains uncharacterized at molecular levels. The information on genetic diversity and structure of this species is required to identify diverse accessions for conservation priority. In present study, we use the transferred SSR markers in this species to study the genetic diversity. The polymorphic markers detected in the wheat. This study can be useful in further genetic analysis of the polygonal verticillum, as both the plant are monocot. 46 SSR primers of the wheat were screened in the pooled sample of the polygonal verticillum, and 26 primers amplified successfully. The markers were validated in 22 accession of the polygonal vertex data, which are collected from the different locations of the northwestern Himalayan regions. And the list of the accession characterized in the present studies, you can see that the samples of the polygonal vertex data which were collected from the different northwestern Himalayan regions. And you can see the profile of the 38 wheat SSR primer in the pooled samples of polygonal verticillum. There's amplifications of the primer in the polygonal verticillum DNA. And this is the profile of the SSR primer in 22 accessions of polygonal verticillum. And the methodology of this present study, the genomic DNA were extracted and PCR for all the markers were performed in 26 microliter reactions volume. The amplified product was separated on 1.5% agar gel using the 1x TBE buffer and visualized using the ethidium bromide and UV lights. The fragment was scored manually and the data were analyzed by computing various diversity indices, the expected heterozygosity and observed heterozygosity and Shannon information index were obtained using the POP gene version, that is 1.32. Next is the PIC were calculated as per the BOTS 10 et al. Methods. From the screen 40 microsatellite primers pairs, the 10 microsatellites were amplified ambiguously and were used for characterization of 20 different accession of the polygonal verticillum, and average 2.35 alleles were detected which showed low allelic richness. The main value of the observed heterozygosity and the expected heterozygosity was 0.462 and 0.480 respectively. The highest value of the PIC were 0.610 shown by SSR 17, while the lowest was 0.141 shown by the SSR 13 with an average 0.385. These are the sequences of the primer which were used for the amplification works. And of the 12 polymorphic markers, the only 33% markers had a PIC value more than 0.5. Highest 1.22 Shannon information index were detected by SSR 17, while the lowest, that is the 0.37, were detected by the two markers SSR 18 and 19, with the mean value of 0.73. The cross transferability rate of these microsatellite markers was 71.4% in polygonal verticillum. The maximum of the markers developed were also conserved in polygonal verticillum can be efficiently used to study these genetic characterizations. The cross transferability of the newly developed markers also suggests their wider utility for characterization purpose in other related species. These are the dendrograms showing the two different groups of analyzed accession of polygonal verticillum, group 1 and group 2. Structure analysis of 22 accession of polygonal verticillum. Here you can see cluster 1 and the cluster 2. And conclusion of this current study, the result of the present work highlighted the need of the investigation the genetic diversity in various existing population of polygonal verticillum in northwestern Himalayas for identification of diverse genotype and their conservations. The cross transferability transferred microsatellites selected in this study can be useful for various genetic analysis in polygonal verticillum and other related species. Efficient conservation and management strategies are required for sustainable utilization of this plant resources. Thank you.
Hello, Dr. Suresh. Dr. Suresh Kumar ji.
Thank you.
Dr. Suresh, please ask some question regarding the oral presentation.
Dr. Suresh Kumar ji.
Dr. Rajesh.
Yes, Pitambar.
Oh, yes, sir.
So you have worked a lot on this plant and molecular techniques you have applied. Uh, what is the first of all, what is the significance of this plant in relation to Himalayan region, and how much is the contribution in the edibility or pharmaceutics?
Yes, sir. This plant is used generally for the tonic purpose. Basically, especially for the this plant has a aphrodisiac property, and is also used against the tuberculosis and hyperglycemia. And there are so many other reports where the plant has been utilized.
Okay, fine. So, what is the current status of this plant in terms of vulnerability or its distribution among Himalayan region?
Yes, sir. Actually, this plant is vulnerable in this Himalayan region due to the anthropogenic activity, and lavishly this plant is being utilized by the local people for the uh, this medicinal purpose.
Okay. Okay, fine. Thank you.
Thank you, sir.
ओके पतंबर जी
Thank you, sir. Thank you.
Okay, Parvinder, so are you ready for the presentation? Parvinder, so
मैम मे बी सिग्नल प्रॉब्लम, मैं कोशिश कर रही हूँ मैम, 2 मिनट।
okay.
मैम अब विजिबल है?
नहीं, अभी भी स्क्रीन शेयर नहीं हो रही है आपकी।
नो मैम।
No.
शेयर स्क्रीन।
विशाल भाई ने join किया।
दीप्ति है कोई? परमिंदर, जब तक आप अपना सेटअप देखो।
So let me consider.
ऐसे बोलना था। Sir, Priyanka Thakur is there.
Okay, you have to start.
हाँ जी, प्रियंका ठाकुर।
Yes, sir. Can I start?
हाँ जी, हाँ जी, अपनी स्क्रीन शेयर करो जल्दी।
Okay, sir. Okay, okay.
लगा रही है टिकट? पेन के साथ, पेन के साथ ही
नहीं?
Sir, is my this screen
हाँ
is
जी, हाँ
visible?
जी, विजिबल है। चलिए आप।
Okay, okay, sir. Shall I start? Hello.
हाँ जी, हाँ जी, आप स्टार्ट कीजिए प्लीज।
Okay, sir. Okay, okay, okay. Sir, my topic is ethnobotanical importance of Bauhinia variegata. So if I will talk about the plant Bauhinia variegata is a deciduous flower-bearing tree, and it belongs to family Fabaceae. It is distributed in tropical and subtropical region of India and world. This plant in Sanskrit and Hindi is known as Kachnar, and the meaning of Kachnar is a beautiful glowing lady because of its very beautiful flowering pattern. So this is the photograph of the tree in its wild habitat. So this plant generally grown in wild habitat, and this plant is also cultivated at the banks of the road uh, and in parks for its ornamental purposes. The stem of this plant is woody, erect, branched, and cylindrical. And if I will talk about the ethnobotanical uses of this plant, each and every part of this plant is very useful. So if I will start with the bark, the decoction is prepared from the bark of this tree, and which is used for the treatment of various diseases. Like the various medicines are prepared which are used to treat blood pressure, diarrhea, dysentery, like diseases. And if I will talk about the leaves of this plant, so leaves are petiolated, bilobed, and it resembles to the shape of a camel foot. So that's why this is also known as camel foot tree. So the leaf extract has many properties, many antimicrobial properties are there, many antifungal and antibacterial properties are there. So they are used to cure the disease like jaundice, stomach tumors. Are of this tree, the flower is white, and it possesses for the treatment of pile, preparing the raita, pickle, curry, and pakoda preparation. And generally they are also used for the feeding of these milting animals. Seeds of this plant is also very useful. They are the source of carbohydrate, fats, fiber, and